When you think about a key, you probably picture something rigid.
Right, like a standard brass house key.
Yeah, exactly. A brass key cut perfectly for one very specific lock.
It's a fixed shape designed for a fixed mechanism,
Mm,
which is, predictable and honestly a little limiting when you think about biological systems.
right. But imagine a thief who doesn't bother carrying a heavy ring of keys. Instead, they just carry a vial of liquid metal.
Oh, that's a cool way to think about it.
Yeah, so they pour it into any lock, and the liquid instantly senses the exact microscopic shape of the tumblers. And then it just hardens into the absolute perfect master key.
Wow.
And that is essentially what the marine cone snail does every single time it hunts. It fires a harpoon loaded with this chemical liquid key that perfectly molds to the victor's nervous system.
It's terrifying, but also kind of brilliant.
It really is. So, welcome to today's deep dive. We are looking at an absolutely fascinating stack of sources today. We're pulling from peer-reviewed neuroscience papers in the Journal of Neuroscience and Marine Drugs.
Quality sources, yeah.
Plus, this highly specialized database called Conoserver, and even a couple of really heavily debated YouTube clips.
It is an incredibly eclectic mix of information.
Yeah.
But, there is a very clear thread running through all of it.
Which is?
To understand the cure, you first have to understand the killer. I mean, nature's most terrifying poisons often become our most precise medical tools.
I love that. So our mission today for you listening is to unpack exactly how the venom of the marine cone snail, specifically these things called conopeptides, interacts with the human nervous system.
Right, we're really getting into the biology of it.
Yeah. We are going to look at how they target specific ion channels and dictate the release of neurotransmitters.
It's all about how this ancient biological weapon connects to cutting-edge pain relief.
Exactly. And ultimately, we'll see how it ties into some massive modern controversies about human health and nicotine.
But to understand how the human brain gets hacked, we really have to look at the hacker itself first, right, the snail. These marine cone snails are predatory gastropods. So they don't just slowly slide over their prey like, a garden snail.
Thank goodness.
Yeah, right. They have evolved this highly specialized hunting mechanism. They actually use a hollow, hypodermic needle-like harpoon.
Which the sources said is actually a modified tooth.
Exactly, modified radula tooth. And they use it to actively inject venom into fish or worms or even other snails.
That's so wild. I read in the sources that this venom gland is basically a pressurized pump.
Yep.
So they fire this microscopic harpoon, pump the venom in, and the prey is just instantly paralyzed. But the venom isn't just one simple poison, is it?
Oh, not at all. It's a massive, mind-bogglingly complex cocktail.
How complex are we talking?
Well, if you look at Conoserver, which, by the way, is an entire dedicated biological database just for tracking these specific peptides, it lists over 8,500 distinct protein entries.
8,500, wow.
Yeah, and that's across 126 different species of cone snails.
See, there was a statistic in the research that really caught my eye regarding that. Out of all those species and all those thousands of venom compounds, there's only an estimated 5% overlap in venom components between the different species.
The diversity is just staggering. I mean, the active ingredients doing the heavy lifting here are called conotoxins.
Okay.
And they are a type of conopeptide. Physically, they're very small, usually just like 10 to 30 amino acids long.
That's tiny.
It is. But they are structurally incredibly robust because they are heavily cross-linked with these strong disulfide bonds.
Right, the sources called them disulfide rich.
Exactly.
Yeah.
You can think of those bonds like molecular armor. It holds the peptide shape together so it doesn't just degrade in the ocean water or inside the victim's bloodstream.
The papers also mention that the snails use this biological mechanism called variable peptide processing.
Yes. That's how they rapidly evolve and expand this huge venom library.
So they are constantly tweaking their own genetic outputs to make new venom variations.
Basically, yeah.
Going back to our earlier thought, it's like a computer hacker generating millions of unique structural passwords rapid fire, just hoping one perfectly fits the target's firewall.
That is a great analogy.
Yeah.
And that biological messiness, it's an intentional evolutionary feature.
Vanilli. Intentional how?
Well, by constantly shifting their peptide structures over millions of years, cone snails have inadvertently provided modern researchers with a massive natural combinatorial library.
Like a catalog of drugs.
Exactly. We're talking about over 50,000 potential neuropharmacologically active compounds.
50,000.
Yeah. And the crazy part is that these compounds happen to be hyperspecific to mammalian membrane proteins.
Okay, 50,000 potential compounds that just happen to fit our biology perfectly.
Right.
So how do these snail-generated passwords actually unlock our nervous system? Because, I mean, the precision here is what makes this so important for neuroscience, right?
Absolutely. The primary mechanism is modulation. Conotoxins basically modulate the activity of human ion channels,
Like which ones?
specifically sodium, potassium, and calcium channels, as well as ligand-gated ion channels.
And just to clarify for everyone listening, these channels are essentially the physical gateways that allow electrical signals and chemical messages to travel through our nerves.
You nailed it. They are the gateways.
Okay, so the papers highlight one major group of these toxins called alpha-conotoxins.
Yes, the alpha-conotoxins are fascinating.
And they specifically target neuronal nicotinic acetylcholine receptors, which the papers abbreviate as ARARs.
Right.
The sources note they have a very specific structural framework. It's written out in the text as CCXMCXMEC. Can we translate that alphabet soup for the listener?
Yeah, let's visualize it. So the C's stand for cysteine residues.
Okay.
Those are the anchor points that form those tough disulfide bonds we talked about earlier.
The molecular armor.
Exactly.
Yeah.
And then the XM and XN, those represent the loops of amino acids that are strung between those anchors. Yeah, imagine a suspension bridge. The C's are the concrete towers, and the X's are the lengths of the bridge suspended between them.
Oh, I see. And the literal size of those loops, like the length of that bridge, whether it has a 47-loop structure or a 35-loop structure, that dictates exactly which receptor subtype the toxin targets in your brain.
That is the absolute key to all of this. By changing just one or two amino acids in that loop, the snail basically files down the shape of the key.
Wow.
Yeah, allowing it to target exact receptor subtypes and block very specific neurotransmitters from being released.
The Journal of Neuroscience papers give us two brilliant examples of this, actually.
They do?
First, there's alpha-conotoxin MII, which they originally isolated from the Comias magus snail.
Right.
And it perfectly binds to what's called the alpha-3 beta-2 receptor interface. By plugging that specific lock, it selectively blocks the release of dopamine.
Dopamine, of course, being the critical neurotransmitter for reward, motivation, and motor control.
Exactly. And the paper mentions this toxin is incredibly potent. It has an IC50 of 0.5 nanomolar. Yes.
What does that actually look like in practical terms? Because 0.5 nanomolar sounds small, but I can't really picture it.
To put 0.5 nanomolar into perspective, imagine dropping a single grain of sand into an Olympic-sized swimming pool.
Okay.
And having that microscopic concentration be enough to completely shut down the pool's entire filtration system.
Are you serious?
Yeah. It takes an impossibly tiny amount of this venom to completely lock up the brain's dopamine receptors.
That is terrifyingly efficient.
It really is.
And then the sources look at a totally different toxin, alpha-conotoxin AOIB, which they got from the court cone snail.
Right, the court cone.
This one targets the alpha-3 beta-4 receptors, and by hitting that specific lock, it selectively inhibits the release of norepinephrine, but it leaves dopamine completely ineffective.
Perfectly isolated effect.
Let me just make sure I'm fully grasping the scale here. By just changing the physical size of a microscopic loop in the peptide, the snail dictates whether it shuts off your dopamine or your norepinephrine.
It's like having a master key that can be filed down by a millimeter to specifically open the joy door or filed a totally different way to open the alert door.
That precision is exactly why neuroscientists treat these toxins like absolute gold.
Because they're useful tools.
Exactly. You have to remember that human brain receptors are notoriously homologous. I mean, they look structurally almost identical to each other.
Oh, so they're hard to tell apart.
Very. For decades, it was incredibly difficult to tease apart exactly what each receptor did because our synthetic drugs were just too clunky.
They just hit everything at once.
Right, they would hit multiple receptors at the same time. So without the extreme precision of these alpha-conotoxins acting as chemical probes, it would have been nearly impossible to definitively prove that alpha-3 beta-2 receptors primarily control dopamine release, while the alpha-3 beta-4 receptors control norepinephrine.
That's wild. We literally mapped our own brains using the hyper-evolved weapons of a marine snail.
Nature is pretty incredible that way.
But this brings up a massive question for me. If these toxins are the perfect keys, can we use them to lock doors we actually want closed? Like severe chronic pain.
Oh, absolutely. Conotoxins are actually a massive area of focus in pain research for exactly that reason.
Really?
Yeah, the most famous success story is omega conotoxin MVIIA. Pharmacologically, it's known as ziconotide.
Ziconotide.
It directly blocks N-type calcium channels, and it is currently an approved non-opioid painkiller for severe chronic pain.
That's amazing. But the sources detail a really fascinating twist with another compound, right?
Mm.
Alpha-chenotoxin VC1.1.
Yes, the VC1.1 story is wild. So VC1.1 and another peptide called RG1A were put into development as potential treatments for neuropathic pain.
Okay.
Initially, the scientific community thought they worked the standard way by blocking those alpha-9, alpha-10 nicotinic receptors we just talked about.
But the research took a sharp left turn. They started testing post-translationally modified versions of VC1.1.
Right.
And just to clarify that term for you listening, post-translational modification basically means the snail builds a base protein and then slaps sort of aftermarket parts onto it before firing it off.
Yeah, like adding a sugar molecule or tweaking an amino acid just to change its function.
That makes sense. So researchers tested one of these modified versions called VC1A.
And in the lab, it perfectly blocked the alpha-9, alpha-10 nicotine receptor.
Great.
But when they put it into rat models, it completely failed to relieve any pain.
Wait, let me get this straight. Scientists made a drug from snail venom, saw the original version cured pain, but completely misunderstood how it was doing it.
Yep.
They thought it was hitting the nicotine receptor, but it was actually sneaking in somewhere else entirely.
Exactly. It was using a backdoor.
A backdoor.
Yeah. It turns out the original VC1.1 inhibits high-voltage activated N-type calcium channels, specifically KV2.2, but it doesn't do it by hitting the calcium channel directly.
Then how does it do it?
It acts as an agonist for G-protein-coupled GABAB receptors.
Okay, let's break that mechanism down a bit. An agonist is something that activates a receptor, right? Like turning the lock to the on position.
Exactly.
How does turning on a GABA receptor actually stop pain?
Well, think of the nerve cell as a house.
Okay, a house.
And the calcium channel is the main power line, sending the pain signal up to your brain.
Got it.
Researchers thought the snail venom was just cutting that main power line. But instead, the venom was actually going to a side window, the GABA receptor activating it, and that receptor was sending an internal signal to unplug the calcium channel from the inside.
Wow.
So the nerve's physical pain signal literally cannot fire and travel up the spinal cord.
And they proved this because when they applied antagonist compounds that specifically jam the GABA pathway, like baclofen or pertussis toxin, the pain-relieving effect of the snail venom just completely vanished.
Right, because the side window was locked, so the venom couldn't get in to cut the power.
This highlights something really beautiful about the scientific method, honestly.
Oh, for sure. In biology, getting the right result for the wrong reason happens all the time.
Yeah.
But figuring out the true mechanism in this case, discovering the GABA pathway's role, is a massive breakthrough.
Because it opens new doors.
Exactly. By understanding exactly how that back door works, we open up entirely new avenues for developing chronic pain treatments that completely bypass the devastating addiction risks associated with opioids.
That is so important. So the cone snails have spent millions of years evolving peptides that perfectly target our nervous system, particularly these nicotinic acetylcholine receptors.
Yes.
And the fact that the word nicotine is literally in the name of the receptor is just the perfect bridge into the final section of our source stack today.
It really is.
Because these exact same receptors are currently at the center of some of the most heated debates in public health right now.
Yeah, the receptors targeted by the lethal snail venom are the very same receptors activated by nicotine and tobacco.
Now, I want to take a quick pause here for a second because we're about to talk about nicotine and COVID-19.
Yeah, we need to be clear here.
Exactly. Just to be completely clear with you listening, we are looking at some heavily debated YouTube clips from our source stack today. We are absolutely not taking a side.
Right.
We are not giving medical advice about nicotine or smoking, and we definitely aren't endorsing any public health policies.
Right.
We are strictly just exploring the science behind why these specific receptors are such a global battleground right now.
And the biological context is what matters here. When you understand the receptor, you can understand the controversy.
Oh, so let's look at the sources. There's a clip from Dr. Farsalinos, who is a cardiologist and tobacco harm specialist.
Right.
Early in the pandemic, he observed what he called a COVID-19 paradox. He noted that while roughly 26% of the Chinese population smokes, data from Chinese hospitals showed that only 6.5% of hospitalized COVID-19 patients were smokers.
And based on that statistical discrepancy, he hypothesized that nicotine itself might have therapeutic benefits or maybe somehow block the virus from taking hold in the body.
But he was separating nicotine from smoking, right?
Very carefully, yes. He distinguished the chemical nicotine from the physical act of smoking. But He heavily criticized countries like South Africa for banning cigarette sales during the pandemic lockdowns.
Which brings us to the counter-argument from South Africa's Dr. Delemi Zuma.
Right.
She defended the cigarette ban by pointing out the behavioral and physical risks. She argued that the physical act of sharing cigarettes defeats social distancing entirely.
Which makes sense.
Yeah. And beyond that, smoking inherently compromises the respiratory system, which makes patients exponentially more vulnerable to a severe respiratory virus.
And her argument perfectly aligns with the conventional medical consensus regarding smoking and lung tissue damage.
Exactly. But then we have a completely different angle from a Dr. Brian Ardis.
Oh, right.
In his clip, he claims that nicotine is actually a powerful anti-inflammatory and a curative agent for things like brain fog and long-term cognitive dysfunction. He also points out that nightshade vegetables like tomatoes and eggplants contain naturally high levels of nicotine.
They do.
But why would a chemical found in tobacco theoretically act as an anti-inflammatory in the brain?
Well, if we connect this back to what we learned from the cone snails, the biology actually makes a lot of sense.
How so?
Nicotinic acetylcholine receptors are not just sitting in your lungs waiting for cigarette smoke. As the neuroscience papers we review point out, these receptors are ubiquitous.
They're everywhere.
Literally embedded all over your body.
Right. The research mentions they are present on immune B cells, and they are abundant in midbrain dopaminergic regions, which are the parts of the brain that control pleasure, reward, and mood.
And this is the crucial takeaway, because these receptors are distributed across so many different critical bodily systems, from your immune response to cognitive function to the autonomic nervous system. Any compound that binds to them is going to have profound systemic effects. If you hit a receptor on a macrophage, it might lower inflammation. If you hit one in the midbrain, it releases dopamine.
So it's just doing a million things at once.
Exactly. And this explains why the health impacts of nicotine are so incredibly complex and widely debated.
It's acting as a master switch for multiple systems at the same time.
Yes. And bringing it full circle, it explains why a marine snail evolving a venom that perfectly targets those exact same receptors can be so universally lethal to its prey.
Because it hits everything.
Right. The snail isn't just shutting down one organ. It's hitting the master control switches for the victim's entire nervous system.
That is just incredible. To summarize this whole journey we've been on today.
It's been quite the ride.
It really has. We started deep in the ocean with a carnivorous snail firing a hollow harpoon loaded with a constantly mutating cocktail of liquid keys.
Yeah.
And we learned how those specific kind of peptides, those tiny armored molecules, became the ultimate chemical probes. They allowed neuroscientists to map exactly how human dopamine and norepinephrine release works.
And then we explored the pain relief paradox, where a venom peptide thought to be a simple nicotinic blocker turned out to be sneaking through a backdoor GABAB calcium channel pathway.
Which was a total accident.
Totally. But that biological misunderstanding ended up offering genuine hope for non-addictive pain relief.
Right. And finally, we looked at how those very same receptors are the battleground for global health debates, showing up in discussions about everything from viral transmission to cognitive inflammation.
It's fascinating.
To be truly well-informed, you have to be able to see these invisible threads, the threads that connect the hunting strategy of a marine gastropod to the darkest mysteries of human neuroscience, to the public health debates happening on your evening news.
It's a great reminder that biology is deeply interconnected. The solutions to our most complex medical challenges often lie in understanding the precise mechanisms of nature's most efficient predators.
I couldn't agree more. Which leaves me with one final thought for you to mull over.
Okay.
We talked at the beginning about a liquid metal that shapes itself into the perfect key for any lock. Right. If a slow-moving ocean snail has spent millions of years evolving a venom perfectly shaped to unlock the specific nicotine and dopamine receptors in our brains,
Yeah.
what other deeply specific biological keys are swimming around down there in the darkest, unexplored parts of the ocean, just waiting to unlock the remaining mysteries of the human mind?
Ooh, that is a fascinating question.
Something to think about. Thanks for joining us on this deep dive.